And it appears that we are officially live. Hello, everybody, and welcome to another session of our Sussex Vision Seminar Series, as always within the Worldwide Neuro Initiative. I'm George Kafedis, a master's graduate from Thomas Euler's group, and currently a PhD student with Tom Baden. And as your host for today, I would like to once again begin by thanking Tim Vogels and Panos Bozelos for putting forward this ever-expanding initiative towards a greener and much more accessible seminar world. Having said that, allow me, of course, to get back to the reason we all gathered here for today and introduce our guest from UCLA, Professor Yirong Peng. Yirong studied biology at Nankai University and did her PhD in neurobiology at the lab of Xiang Yu at the Chinese Academy of Sciences in Shanghai, focusing at the time on homeostatic regulation of synaptic strength during development. In 2012, she joined as a postdoctoral researcher the lab of Joshua Sainz at Harvard, where things became definitely more retina-oriented. In 2020, she started her own lab at UCLA and has remained there ever since, nowadays as an assistant professor in the Department of Ophthalmology at the Stein Eye Institute. In her lab, they employ state-of-the-art omics approaches, genomics, transcriptomics, proteomics, to molecularly describe different retina cell types in both health and disease, from regional specializations within the same retina to evolutionary scales going back hundreds of millions of years. Therefore, today, personally, I'm so excited, and we all have the pleasure of hearing about their latest and I'm sure exciting findings in her talk entitled Molecular Characterization of Retinal Cell Types, Insights into Evolutionary Origins and Regional Specializations. So without any further ado from my side, please all welcome Professor Peng. Yirong, the stage is officially all yours.
Thank you, George, for the introduction. And I want to take 1 minute to thank you, George and Tom, for inviting me to present at the Worldwide Neuro, the Sussex Vision Series. This has been an excellent platform for us to really learn about the different aspects of visual system. So today, I'm extremely delighted, honored to present our recent findings. So let me start by sharing my slide. Okay, let me see. Is all good, George?
Yes, I can see the whole slide, I think. I can confirm also when we go to the next ones.
Okay, cool. So today my talk is going to focus on the molecular characterization of the retina cell types. And with that, we hope to provide the insights into evolutionary origins and regional specialization. So title may be a little bit abstractive because without the context, but I want to show this image of collective eye to show you how diversity of form of the eye shape across different species. So in that case, we can imagine even different species may share the same environment. The way they perceive the surroundings may be completely different, largely due to the different forms of eye, and most importantly, the retina lying in the back of the eye. So for this community, I don't think we need further a lot of details about the retina. And some of you may use two of these popular animal models. One is the mice represent the mammalian system, another is the zebrafish for the non-mammalian vertebrates. So the common retina feature between these two species is the 3 layers retina. We all know they are photoreceptors located in the outer nuclear layer, and the individual interneurons such as horizontal cell, bipolar cell, amacrine cell located in the inner nucleus layers, and the output neuron retinal ganglion cell located at the back of the outer retinal ganglion cell layers. And the cell classes form synapse between the 2 synaptic layers, which are the outer proximal layer and the inner proximal layers. But we can immediately appreciate the difference between these 2 model organisms is, for example, in this different color to show the zebrafish largely is a cone-dominant retina. They have so many different types of cone photoreceptors, but the mouse is a ROS-dominant retinas with accumulated rod photoreceptors. But these differences is not a major dividing between the mammalian vertebrate or mouse mammalian or mouse system is largely due to their ecological habitat. So even in the mammalian, we have species can form a cone-dominant retina. For example, the ground squirrel is largely cone photoreceptor dominant for the retina. And in the primate, we do have a certain region is also accumulate with cone photoreceptors. In the other hand, the retina can be also take a lot of adaptation to become a very simplified retina. For example, the anglerfish living in the deep seas, their retina is purely composed of the rod photoreceptor and with a very simple system circuit trying to capture the dimmest photons they can capture in the deep sea. So we also have outer achievers like the eagles. They have extremely developed retina with so many types of photoreceptor and extremely elaborate the interplexon layer. And also we have outliers. For example, these chameleons, their individual eye can focus independently and they use a convex lens completely allow them to see 330 degrees of the visual field. So using this image, I just want to demonstrate one point. They are profound adaptations in the cellular composition of the vertebrate retina. So the question really interesting for me is, what is the evolution origins of this cell type diversity? In addition to this complexity, we also, even within the same species, the retina cell type accumulated are different in different area of the retina, which form the regional specialization. For example, in the primate or the bird of the prey, they have the special fovea structure, which are formed with a concentrated cone photoreceptor and retinal ganglion cell, which allow them achieve the max resolution and the spatial resolution and chromatic resolution. So the second question I'm interested is, how is the regional separation formed with the different cell type? So to address these questions, we have to go back to the fundamental approach. It's how we can describe or characterize the cell type in the vertebrate retina or generally in the retina. So this is a long continuing journey and starting from a century ago from the Roman Cahal, which used the gauge staining, to really looking at the retina cell type in different species and starting to wonder why they form different shape and what's the function it is. In modern days, particularly from Josh Sands, we know he spent over 2 decades trying to build up the transgenic mouse lines from the famous Sci1 line, the Brimble, and other GenB line, trying to really characterize different cell types in the mice retina. And this genetic approach also widely used in the zebrafish. So then when in the recent years, Josh quickly realized there may be a much more efficient way to do it, which is the leverage combine the a transcriptome sequence with the high throughput, the microfluidic system, particularly with the great computational scientist Karthik Shankar joined the group. We quickly can, using the single cell RNA sequence to generate the cell atlas from multiple species. So let me briefly introduce the single cell sequence methods. Basic is you dissociate the retina structure to either the individual cell or nucleus, and you can either go through the FACS enrichment sorting to your specific interest population or just go through the microfluidic. So basically you generate the droplets contain individual cell and trying to analyze their gene expression in individual cell. Through the computation, we form clusters of the cell, and the individual cluster will define as a cell type. So I was lucky enough to be participating in some of this research and allowed us to identify the cell types in many primate, mouse, and chicken feral species. So To validate the single cell sequence, we did spend a lot of efforts trying to use the in situ method or morphology method to corresponding the type we verified with the transcriptome result. But this still has concerns with this method is because we generally dissociate the retina, which means individual cell is isolated, well truncated their dendron and axon, and you only focus on the mRNA from their somatic site. But it's reassuring enough is the whole retina community is starting to take a series of a data set and trying to validate whether our molecular characterization is real. Particularly, we are very excited to find the recent publication from the Teresa lab, who really identify the primate, the retinal ganglion cell type 10 are indeed on the SGCs. So this really kind of encourage us to really continue into this molecular characterization, really identify the cell type from different species. But of course, the molecular characterization is not the full pictures. There are so many different population of function methods has been generated and improved, which are including the calcium image, electro image, and adaptive optics. Particularly, many of these methods trying to align with other characterization properties like e.g. Chichilikis group can really register electrical signals with the morphology, and also David Williams group trying to use the optogenetics and the adaptive optics to do the simultaneous function activation and function recordings. Particularly the great efforts from the Greg Schwartz group, they have a very ambitious goal to really align the morphology, molecular function, and the connectivity together. So we can imagine if one day every species be categorized in these so many different dimensional, we can quickly solve what exactly the revolution mean to provide individual species with so many diversity of cell type. But at this kind of stage, looks like the transcriptome method is still most efficient of feasible ways to really sample the cell type from different species. For example, we can learn from the evolutionary studies is how they can call the homology or the convergent evolution or concentrated evolution, just treat individual gene profile of individual cell type as a phenotype. Or later, we can call the functional type as a phenotype, then really build up the evolutionary history of how the cell type build up in individual species. As one example is the great work from the Karthik and Josh. They really using the single cell sequence to characterize the cell type from 17 species. Among them, they identify, among them, there are 13 mammalian species. So the striking finding they find is among the 13 mammalian species, they can successfully integrate individual types to form an ortholog type. Then they can build up the homolog relationship among all these mammalian systems, which really provide an evolutionary history of how the cell type is generated in mammalian system. But of course, this has some challenge, which we'll see here is if we step out the mammalian zones looking at outside the mammalian vertebrates, the corresponding is kind of decline. So this kind of common challenge to using the transcriptome method to really relate cell type across a long phylogenetic distance is largely, there's a lot of changes in the chromatin reorganization and also have some whole chromatin chromosome duplication, particularly in the Teleostes species. So in this talk, we want to show our two recent studies to provide some additional information or evidence to hopefully we can provide insights on the evolutionary origin of the diverse cell type in vertebrates. Here, particularly, it's a study of the sea lamprey. Another thing is we want to provide evidence is or provide some mechanism to really start to thinking how the developmental formation of the regional specialization happens. Here we use the study of the common marmoset. before I share the result, I really want to attribute this acknowledgement, the efforts from my 3 lab members, Junqiang, Ling, and Martina, who is taking the leading roles on this project. So without them, we cannot get this far. So first of all, I will talk about the result from the Lamprey. So when I started my lab in 2020, I joined the Stein Eye Institute, and my 2 colleagues approached me, Gordon and Sam. They said, okay, Yirong, you are very good at doing the single cell. There's a species we have in the lab. So they introduced me this exotic species, the lamprey. That's my first time in my life to see such a creatures, although I heard the name, but never see it in person. So the special about the lamprey is it represents the cyclostome, which means jawless. But for the rest of the nascent stone, which is all the rest of the jawed species, which has a jaw. For example, the golden Sam has a beautiful smile because they have the jaw. So the specialty about the lamprey is they diverged from all the rest of the jaw species around 500 million years ago, which is almost the time the emergence of all the vertebrates. So we think if we can compare the lamprey with the rest of the jaw species, they can tell us something about the evolutionary region of the cell type. So a little bit of caveat about the lamprey retina or eye is it is kind of starting from the immature eye shape. For example, in the lamprey's larva stage, which we call the ammocyte, which is only have a tiny eye spot, high hidden under the skin. But during the metamorphosis, they will change their morphology of the face shape, but also the eye starting to regenerate. And then in the adult stage, they form like the mature eye and the retina as the rest of the jawed vertebrae. So they still have major difference between these 2 species, although they all form a 3 layers retina structure. For example, in the bottom is a retina structure which we all recognize is shared with all the jawed species. We have 3 layers of the cell type, and with the retina ganglion cells sending the axon to the brain through the neurofiber layer, which is located in the most inner part of the retina. But the retina of the lamprey is slightly different. Most of the retina ganglion cells are located in displayed position, and the exit route of those retina ganglion cells is through the middle of the retina. It's kind of above the inner plexus layer. So we think that's kind of interesting, and if we can quickly get the single cell sequence of a lamprey, maybe it's kind of a low-hanging fruit for my lab to really have a quick publication soon. But then we run into a first bottleneck is, although we take so familiar with different model organisms because they all have very good or complete genome annotation. But when we're looking at the sea lamprey, we find is, although the genome is all be sequenced, but they have a large population of the gene without gene symbol. So that means compared to the mouse models, almost all the genes be annotated. So that means if we want to compare the lamprey with other species, we may end up don't know what kind of gene we're looking at or compared. So we have the first step is trying to clear up all these kind of unannotated genes by looking their protein product name, but also use the finder to compare with other species. So that's for the great effort we did to update the genome annotation of the sea lamprey. But another thing we did is because all the gene annotation generated from the lamprey is either from the germline of other part of the body, is not exactly from the retina. As we all know, retina has special genes, for example, rhodopsins. So trying to improve the annotation file, we also use the TrueSeq, which is using a long-read sequence to analyze the RNA sequence specifically from the retinas to really improve the annotation accuracy. So as an example of using the rhodopsin genes can show the bottom one is from the NCBI, original annotation of the rhodopsin. But with our choosing, we largely improve the annotation. So by identifying new 5' UTR or additional UTR in this gene. So this will improve our mapping rates by using our own generated retina specific transchrome compared to all the existing annotation file. So with all these efforts by updating the genome and also updating the gene symbols, we largely improve the data count matrix we can generate after single cell sequence. So with that, we feel comfortable we use this updated annotation file to really understanding the lamprey. But because still the lamprey is a new species for us, and we really want to take a careful step to even determine what we call a cell class in the adult sea lamprey. So we do the classification slightly different than the previous way. Is we first use a low PC number for the reduction, trying to find the major relationship of the cells, individual cells, and assist with a single R annotation. Let me explain a little bit about that. The 6R is kind of reference method is we first want to identify what could be the authentic cells in the lamprey by combine the gene expression and in situ validation and use these authentic cells to generate a reference to calculate all the rest of the cell in the lamprey. With that, we can comfortably say, okay, this group are RGC, this group are AC, and that bipolar cell photoreceptor, and so on and so forth. So once we can characterize all the cell classes in the sea lampreys, we can quickly separate the individual cell class and really identify individual cell type in it. So far, we can identify 2 photoreceptors for horizontal cell, 8 bipolar cell, 11 amacrine cell, 4 Starburst amacrine cell, 4 Muller glia, and 41 RGC. So this diversity of cell type is really surprising for us because consider this kind of primitive vertebrate species, they have such a diverse form of cell type. And the least number of cell types beyond we can even validate. So what we want to approach for the next stage is really we want to use the lamprey to compare with other known jeweled vertebrates retinal cell type to generate what could be related from the lamprey to other species. So we chose the 3 species of, for example, the mouse and chicken and the macaque. It's not because they share any kind of ecological habitats or because it's our favorite. It's because the 2 reasons. One is they have the same modality generated as the lamprey, which is a single cell RNA sequence. Second is some of the species, their cell type be well characterized and validate. So our hypothesis, if any shared cell type between the lamprey and any jeweled species could be present, could be identified as common cell type, which means this cell type could be already present in the primitive vertebrate ancestry. So that's kind of our fundamental goal or ultimate goal to do this cross-species comparison. So I will walk through several different classes and the different cell type to tell you a kind of the basic findings we learned from studying the lamprey cell type. So first of all, it's a little bit about the photoreceptor. So basically, the vertebrae all have the duplex retina, which means the separation of the rods and the cones. So usually we think the rods is generated or evolved from the cones, which is based on the lamprey result. So the lamprey has all cones, which they have cone shape, which means they are serious photoreceptor, but they don't have individual separate disc. So it means the opsin is free floating through all the membranes. So from the Gordons is the first group recorded the sea lamprey. They use both the single cell recordings and also probability intensity recording identifies the rods and cones in the lamprey actually has different photosensitivities and the kinetics, which is extremely similar to the mammalian system. So based on this, the hypothesis is could be the rods is originated from the cones. So from our data set, we also find the two photoreceptors, PR2 and the PR1, particularly they are separated by expression rhodopsin and the rhodopsin, which is validated by the in situ. It's most striking for us, if we're looking at all the phototransduction cascade molecules, they kind of completely separate between the rods and the cones, which means determine the function of the rod cones. The system already set up in the lamprey, although their morphology is quite different. And the most striking for us is even the transcription factor determine the rods versus cones. For example, in the mammalian species, we know the NIL and NR2E3 expressed by the rods, they also shared in the lamprey. So the MAF2 is a homolog for the NIL. So that means the conservation of the photoreceptor is extremely similar between the lamprey or mouse, even they separated 5 million years ago apart. So then I will walk through different types. For example, since we find the rods, so we want to know whether we can find the rod bipolar cell in the lamprey. From SAMS and Gordon's, they successfully recorded the on-off bipolar cell. And indeed, we can find 8 cell types of the lamprey bipolar cell and with the separation on bipolar cells. And interestingly, on bipolar cell does not express the GRM6 or M1, but they express the GRM8 and 3. So from this data set, we generate some Pearson correlation to see the relationship of the lampreys bipolar cells and find they are very distinguished group, which are the BC1 and the BC5. And when we're looking at the gene expression, they express something genes we call our attention, which is the PRKACA and the GRMAMD1B. Both the PRKCA is sometimes can be recognized as a homolog of PRKCA, and the GRMD1B is markers for the rod bipolar cell. So indeed, we use the mammalian antibodies against the PRKCA. We can identify the certain type of the bipolar cell in the lamprey species. The morphology quite represent what we see in the mammalian system. And at this same time, so we saw this beautiful work from Takeishi group. They looking at the zebrafish, they find almost the same kind of pattern of two group of the rod bipolar cells in the zebrafish, and then they did a very carefully functional connectivity validation. So this suggests us the BC1 and the BC5 could be rod bipolar cell in the lamprey. So we did further do an integration to compare with the mouse species, which means we integrate the cell from the lamprey and the mice to see which cell type can be integrated together. We find it's the BC1 and the BC5 indeed integrate together with the rod bipolar cell from the mice. And also from the transcriptome mapping based on the tree building model, we can also validate the BC1 and the 5 map to the rod bipolar cell in the mice. So this is quite clear for us, is lamprey may do have a rod bipolar cell, and quite similar to the zebrafish, they may have the 2 bipolar cell type as well. So the next is a horizontal cell. We find the 4 group horizontal cell, and they are both have the melanopsin expression. And the 4 group can separate to 2 subcluster. One is the LHX1 positive one, and the other is the LILO1 positive one. So because the mice only have 1 horizontal cell, so we integrate with the chicken one, which has 5 horizontal cells. So when we do the integration, we find they form a 2 kind of cross the major group. And interestingly, we find the lamprey group, which are divided by the LHX1 or ILO1, are specifically integrate with the chicken's type 1 horizontal cell or type 2 horizontal cell. So that means the different horizontal cell may already is set up in the lamprey species. So the next cell type we're interested in to really looking at the starburst cell, which is a large population in the lamprey cell population, and particularly the starburst cell is the on-off circuit in the inner proximal layer. So from the lamprey, we identify 4 starburst cell type, and they all expressed chat. And we can see some located in the inner proximal layer, some located in the GCL layer, could represent the off stubborn cell or on stubborn cell based on somatic position. And quite striking is all the stubborn cell also express MAGAF10, which we know is transmembrane proteins mediate the mosaic organization of the stubborn cell. For me, it's extremely exciting is I find the two stubborn cell are highly expressed the FASF2, which is reminding me the mouse stubborn cell with the on stubborn cell specifically express the FASF1. And FASF1 is important to determine the fate of on stubborn amacrine cell. As we showed before, if we knock out the FASF1 in the mice, all the on stubborn cell become off. So that means the lamprey may already have the on and off grouped stubborn cell, and their cell fate could be determined by the same family member of the FASF2, which is quite similar to the mice. So from all these presentations so far, we kind of surprised by how conserved these two species are, or the lamprey with many of the jaw species. But also the question get more complicated is we identified 41 type of retinal ganglion cell in the lamprey. So how we're going to understand that? So there's many kind of retrograde labeling in the lamprey to identify 8 group of the RGC type based on their location and the central target. So we're trying to do a little bit kind of relationship, kind of clustering, and we can group them to maybe 7 clusters. Some unique feature we can find is certain group are enriched by express FOXP1 and FOXP2 gene, or some group express the OPN4 gene, which could suggest then they are IPRGC. But we cannot do any more for like validation or recording to verify which one is which with the time concern. So what we did, we turned into the mouse system, which has well-characterized retinal ganglion cell type, and some of them be validated by function and morphology. So we're trying to do the species integration, which is a challenge because they are very diverse in both species, and the gene expression difference is huge between these two species. But our ultimate goal is trying to say is whether we can try different integration methods to really form an integration map, which will largely preserve the species information in one or the other species. So we did that. We think is kind of the best integration we can get. Is in this integration map, we can also preserve the lamprey retinal ganglion cell type information and the mouse retinal ganglion cell type information. So building on these, we can really see which type of the retinal ganglion cell in the lamprey may closely relative to the mouse retinal ganglion cell. So then we generated the confusion matrix to really looking at the type identity. So surprise for us is we did find several type of the retinal ganglion cell from the mice, actually well mapped with the lamprey, which are including the alpha-off transient cell, on-off DSGC, JRGC, and some W3 RGC, F-medium FGC. So looks like they are conserved in this retina type, may mediate direction selectivity or motion sensor. It's even striking for us is this mapping the type even share the common molecular markers. For example, we know CAD-PT and NRP2 are labeled on-off DSGC. They are both showing in the lamprey and the mice. And so does the FOXP2, which labeled F-mini on. But a surprise to us is we find the IPRGC in the lamprey, which expressed OPN4, but they lack of the corresponding with the mice, which means the IPRGC is a conserved cell type, but how they generate could be a convergent evolution. Largely is due to we didn't find the EMOS or TBR2 expressed in the lamprey, which is determine the fate of the IPRGC in the mice. So they could have some species difference there too. So the last class I want to mention is the amacrine cells in the lamprey. We find the 11 type, but we know in the mice there are 63 type. How we build up the connections Similarly, we generate the integration map to see how well the species information be preserved. So we find, using the confusion matrix, we find the pattern is quite different when we compare with the retinal ganglion cell between these two species. Is we find 30 type of the amacrine cell in the mice could map to maybe 8 group of the AC type in the lamprey. And one of them is interesting is the A2 amacrine cell, which in the mice is AC3. And more strikingly is the markers in the amacrine cells, for example, in the A2 amacrine cell from the CAR2 also present in the lamprey. So that means they could have certain species conservation, but largely individual species divergent in their own IC type. And particularly these divergences is maybe get further diversification in later species, which means in the lamprey, early jaw species, AC2, they could kind of have further functional diversification in later jaw species, although there are multiple to one relationship. Don't know whether I can explain. So with that, we have a great kind of conclusion is the lamprey and the other jaw species, although they diverge in so far away, but they kind of share a lot of cell type similarities. So we want to further understanding is what could be the driving force for these conservations. So trying to turn in these questions, we can think about one way to address it is not looking at the gene expression profile, but think about the protein activity. What that means protein activity is group genes function. For example, if we want to looking at whether the rhodopsin activate, maybe it's more accurate ways to measure the level of rhodopsin. But for the single cell sequence, the method is measure the gene expression profile. So how we can achieve that? So Junqiang from my lab actually training with the cancer biology looks like it's a very well-studied field in the cancer biology. They call the reverse engineering. It's basically trying to infer the protein activity from the gene expression profiling. So what we did is from the gene expression matrix, we want to identify those co-expression network. We call it the regulome. This regulome may be transcription factor or co-transcription factor or signaling proteins or membrane proteins. Any proteins can drive a lot of correlation of gene expression. So by identifying these master genes, we can further generate a statistical model to looking at their downstream or correlated gene expression to reinfer what's their protein activity is. So that's will transfer a gene expression profile to a profile of protein activity. With that, we can identify what's the master regulators in the different species and really compare whether they are functional or gene regulation similarity. So with that, we first tested whether this method can work in both the lamprey and the macaque. Here we choose the macaque as the most different species from the lamprey because the goal is trying to really find what's the extremely conserved master regulators between these two species. So from both species, using the protein activity, we can also successfully cluster individual cell class. And we can nominate many master genes, regulated genes, which also related to their cell class function. For example, we find the GN81, which is indeed the photoreceptor function in both species. So that's proof this method did work and provide us a way to really can identify what could be the key important genes in the two different species. So then we can correlate it which could be the conserved protein or gene network. So with that, we ranking all the master genes from the macaque really find what are shared in the lamprey. So in every cell class, we can identify multiple conserved master regulator between these two species, which means those master regulators in general, the cell classes could largely have the ancestry origin. So it means they already kind of present in the ancestry, vertebrate ancestry. But interestingly, many of these are not transcription factors, but instead they are either signal genes or membrane proteins. So that means to determine a cell or cell class, maybe the ultimate goal is really trying to generate the same function. So using this method, we're trying to say in which cell class are most conserved across the between the lamprey and the macaque. What we did is we rank all the master regulator in the macaque one cells of the one class. For example, here is the mutaglia, and looking at how consistent this ranking is corresponding with the lamprey. So if they corresponding well, they will have a high NES score and a very significant p-value. So with this method, we compared all the 6 cell classes. Surprising to us is we find actually the mutaglia and the photoreceptor are extremely conserved between the 2 lineage, but instead the bipolar cell has a lot of variation between these 2. So this is still part of us. We have not have a good idea to explain that, but could be largely is the difference between the could be the difference between the mammalian or non-mammalian, or could be the just 2 lineage differences. So summarize this study of the lamprey, we hope the insight into the origin of the cell type diversity is the following. So the vertebrate ancestry may have already acquired a diverse pool of cell type. This kind of provide a flexibility or plasticity for individual of further evolved species to pick up which cell type they want to be inherited. And also we can find rods and cones, rod bipolar cell, on bipolar cell, off bipolar cell, type 1 and type 2 horizontal cell, on-off stubborn cell, A2 amacrine cell, and DSGC types in the lamprey, which suggests that their early emergence in the vertebrate evolution. And this conserved cell type highlights the Primate designed retinal circuit just for the rod pathway, on-off discrimination, direction selectivity, or motion sensor. So the pathway for diversification of the amacrine and the retinal ganglion cell may be divergent between these 2 lineage. And we find some common regulatory elements underscore the ancestral nature of the molecular origin governing the real side, determine the retinal cell classes. So this is all our exploration of the lamprey, and that's the conclusion. So I'm going to switch the skill to really looking at a different clade, which is much more closer. It's the mammalian, which is separate from the common ancestral around 200 million years ago. It's not that different. So, and then from that, there are 6,000 extant mammalian species. Among that, 300 are primate. The unique primate is we form a certain special, the regional specialization, we call it the fovea. So that's simplify this kind of phylogenetic tree and focus on the mammalian system. So the fovea formation is very interesting. It's only shown in the primate, but only shown in the dino and support primate, which will starting from the New World monkeys to the humans. So the unique organization of the fovea is they form a super enriched cone photoreceptor region and the retinal ganglion cell. So my previous work in the George Sands group, we extensively trying to compare what make fovea unique, and we use the macaque and the human to really identify all the cell types in the fovea of these 2 species. But we missed the one important species, which is the marmoset, which is kind of in the divergent zone of whether the primate were going to have a fovea or not. So these studies, we focus on the fovea of the marmoset, trying to understand how similar the fovea of the marmoset close to other primate. Additionally, the fovea formation is not just born like that. It goes through a long term of specialization, developmental specialization from the early embryonic stage to the postnatal. Particularly, we focus on postnatal is the fovea functional maturation is happening postnatally. For example, when the baby or a monkey born at birth, their cone photoreceptor actually located only 1 layer in the outer nuclear layer, but they have to go through the migration from multiple layers in the fovea region And also they have dramatic morphology changes in the cone photoreceptor. So we think we also want to looking at what could mediate the developmental changes. So with that, we have to generate this kind of cell atlas, including the adult fovea, adult prophylae, and the neonates fovea and the neonates prophylae. So we want to compare whether the cell type already generate at this age and in individual regions. So we did the transcriptal mappings between the regions at the two different stages. They are extremely corresponding between these two. And also we compare the developmental stage in both the fovea and the prophylae, and all the cell type can be found. So generally conclusion is all the cell type are fully differentiated at each region and at birth. So what is special about the fovea maturation? Step back is we want to first identify whether the marmoset share the same set of the cell type as other primate. So we did the integration of all the fovea cell type from the previous reported the macaque and the human and also newly generated marmoset. We did the integration, looks like they are well integrated, and we can first identify cell class and also individual cell type. And we compare all the cell type and the subclass in these three species. We see certain compositional difference, but almost all the cell type present in the three species. The only difference is marmoset don't have this so-called off-X cell. So it looks like the fovea cell type among the primates are extremely similar, which means they use the same set of cell type to form a fovea structure. So then we really want to understand what makes the fovea really unique during development. So we turn into looking at what the most changes across all the cell type between the different developmental stage at two regions. Basically, we integrate the fovea cell and the prophylae cell for individual class, and looking at how strong of developmental changes in the fovea region for every cell type and compared to all the cell type in the prophylae. So using these characterize the regional score and project back to the UMAP of the cell type, we find a different pattern is that in the prophylae cell, almost most of the cells show strong developmental changes, but in the fovea, its selective cell type show changes. For example, there are only ML cones, mural glia, and on-off midge ganglion cell show dramatic changes, which suggested the maturation levels is focused on these midge pathways in the fovea during the postnatal development, but in the prophylae region is generally development. So we further want to dive into what are mediated those specialized developmental changes. So particularly in the context of the cone cells has just dramatically changed their morphology, and we focus on looking at what could be the gene expression changes associated with the cones. From this GOPC analysis, we really find a lot of GO terms associated with the P0 fovea cones, and many of these biology pathway associated with the morphology signal. For example, there are many genes located in the microtubule or do the exon dendrite transport or the morphogenesis. So this suggested the differential gene associated with the neonates cones could mediate their morphology changes, but how it be mediated? So we turn into looking at whether they have transcriptome programs difference between the fovea and the prophylae cones during the development. We first use the inference method, which is called the cynic, trying to generate the regulation, the regulons by the single cell data set, looking at what could be the top regulators between the fovea cones and the prophylae cone. We really identified there are several top transcription programs are either enriched in the fovea cone and the prophylae cone. For example, the SOX6 enriched in the fovea cone, SOX4 enriched in the prophylae cone. To validate this info, the result, we generate single cell RNA sequence of both the P0 marmoset and also the P0 marmoset in the prophylae region. Similarly, we find the gene activity predicted by the single cell ATAC-seq also show the extremely different gene activity of the SOX6 and the SOX4 in the fovea cones. And also we validate with the in situ result. So this suggested during the developmental, they have distinct transcription profile apply in the fovea and the prophet cone. Those could mediate their morphology changes. But the intrinsic program could be triggered by extrinsic molecules. What those extrinsic cues could come in from. We turn into this kind of image or the diagram to illustrate how the environment of the cones located in the fovea. So from this image, we identified the interesting is in the fovea part, the cones are extremely surrounded with the mural glia. So whether the close interaction between the cones and mural glia could provide some guidance cue or niche for the further developmental changes for the cones in the fovea. So how to approach these questions from our single cell data is we're trying to really build up a hypothesis is they do have interaction between these two cell type, then how we can achieve that. So we turn into this algorithm called the niche net is presumably you have one sender cell, they through the secreted molecules of membrane protein could influence a receiver cell's gene expression. So in this particular case, we can think the mural glia could be the sender cell and the cone could be the receiver cell. And through this program, we can really generate what the ligands can secrete from the sender cell and bind to what the receptor and trigger what downstream genes in the receiver cell. So the first step we want to do is to identify what could potentially secrete the molecules in the mural glia. We find actually a lot of membrane protein or secreted proteins are enriched in the fovea mural glia. Particularly, we validate the one is called CTGF, is only enriched in the fovea mural glia, but not peripheral mural glia. And we can indeed find that there are receptors in the fovea cone site. So what's the gene regulation look like? So we apply the niche net, and this side, the left side, is all the ligands, secreted molecules from the mural glia, and the top part is all the downstream genes show difference in the fovea cone. And we ordered by their GO functions, for example, the actin growth, microtubule organizations, and so on and so forth. So once we do this assay, trying to looking at the regulatory potentials, striking for us is not all the ligands show the equal effect to regulate the GO pathway, but instead, there are 4 top candidates show strongly driven the differential genes in the fovea cones. Of course, in the future work, we need to test what exactly this ligand do to the fovea cones, whether they provide some nutrition or extrinsic cues for the morphology changes in the fovea cone. But our hypothesis is the postnatal maturation of the fovea cone could reside in the special niche that located in the fovea. This could come in from the mural glia or other cell class. They provide this kind of special environment for the fovea cone can get further development or maturation and form their mature circuit. So the insight from looking at the region specialization of the primate fovea is that so all the diurnal and support primate share nearly the same set of the fovea cell type. And the selective the fovea cell type, such as cones, mural glia, midge ganglion cell undergo more drastic development changes than other fovea cell types. And the developmental changes in the fovea cone could potentially be regulated by the fovea mural glia. So this is all our current discoveries. I want to end up my talk using this beautiful image from Tom Baden review papers is there are more diversity of cell species the species we can looking at. In my studies, I only looking at the 2 species at the end of the evolutionary trajectory. We hope we can provide insight to the evolutionary origins and also the regional specializations. But of course, there are more is going to investigate looking at all different kinds of species. Without this, we still cannot draw full pictures how diversification of cell types happens in the vertebrate. So with that, I want to end my talk by acknowledge my lab members. Junqiang, Lin Zhang, Martina both involved in the Lamprey and the Marmoset project, but with Junqiang leading the Lamprey project and Lin Zhang leading the Marmoset project. Of course, we have the other support from the Richie, Ali, and Jun Ivies to really let us to finish this project nicely. I want to acknowledge all the other members of my lab who really provide the essential role for us to be productive and support each other. Particularly, they are so brave to join my lab as a new group. And I want to thank all my collaborators, Sam and Gordon, Ella, to provide the collaborations on the Lamprey project. It's a fun project. I'm really pleased that we get this far. And thanking my previous mentor, Josh, to let me continue the Marmoset project. And also thank you, the Guoping Fan and Chang'e, to really provide a valuable Marmoset source, tissue source. I want to thank all my funding agencies. So this is the picture of our current lab members. We're trying to do a handstanding even after challenge hike. So if you are interested to join my group, so don't hesitate to send me an email. All right, thank you so much. I would like to take any questions.
Thank you so much, Yirong, for this fantastic and information-rich talk from both sides of the vertebrate tree, like from lampreys and primates. Personally, I'm in like great awe of how conserved everything looks to be, like different cell classes. They are all the same in all the vertebrates, and you have also different cell types that match individually. And I was wondering, because both in what you showed today, but I also think this is what Karthik was showing a couple of weeks back, we seem to have a higher diversity in the retinal ganglion cell side of things. So if something is less conserved, is this layer? Do you have any speculation why this might be the case?
So I think from the Karthik's work, if we focus on mammalians, his work beautifully show their high-level conservation in RGC type. So for example, all the mammalian species form this kind of ortholog type. That means like have a group of homologous cell type of the retinal ganglion cell. Even the midge ganglion cell could share across mammalian species.
Right.
Which we think is the cell type present, but in the mammalian species is still diversified, which represent at or reflect at the fraction or the percentage of the cell be predominantly present in any species. For example, we know the primate has a lot of midge ganglion cell, but even mouse has certain similar midge ganglion cell, but extremely low fraction there. So from that, we think if we're looking at the mammalian systems, the cells diversification already present in the mammalian ancestor, if any. But looking at the lamprey and the primate or the mice, we still find conserved cell type, but individually we find the species diversity of the retinal ganglion cell. So how much is really kind of non-mammalian vertebrate specific? How much is the lamprey specific? How much is the primate shared? We don't know. So the best way to really address that, to have a fundamental answer, is we still need a lot of sequence or characterization of those representative species from other class of the vertebrate. Then from that, we can build up the more kind of comprehensive understandings exactly in which level the RGC diversification is different from the non-mammalian was the mammalian.
Right. Thank you very much for addressing this. I'm going to the chat where some questions already appeared. They are all from Tom. Maybe people are waiting to join us in the Zoom room to continue with their own questions. I have already posted the link there. And just as a reminder to our audience, after a first moderation from my end, we will continue offline, so make sure you copy the link. So the first question appearing from Tom is about the amacrine cells. And he says that zebrafish also have quite few amacrine cells, perhaps many amacrine cells is a trait associated with tetrapods or related to vision in air?
the vision in air.
Yeah, so because you have like few amacrine cells in aquatic animals and
Yeah.
more amacrine,
Oh,
if there is a pattern there.
yeah, that's kind of interesting observation. We don't know that. So the same I didn't explain maybe not very well is we think there are really a lot of like multiple to one map if you compare the mouse amacrine cells with the lamprey. So we think it's kind of like the pool of cell, like a pool of molecules to determine the AC cell could be there, but they need further a little bit separate to modules. For example, in the lamprey cells, the AC2 can map to multiple. This could be in the mouse or later species. They are form this concrete or concentrated evolution, which can be a progressions of diversification of amacrine cells from the lower vertebrates to higher. So specific case is we also notice the AC class in the lamprey has both GABAergic glycine, but we know in the mice they separate. So this kind of the example to show is this could be a further divergence of the module to individual cells in the later drought, but although the molecules already present in the
Right.
early vertebrate species. So I think it could be similar observed in the vertebrate, the zebrafish amacrine cell.
And another, maybe asking for your speculation question, because we don't have enough lamprey physiology. Again, question from Tom is, why do we have so many horizontal cells in lampreys? What are they
Yeah,
doing?
I don't know, but actually that's what I heard from Tom is, although we said it's only two type of photoreceptors, we saw it's red opsin because we cannot the lamprey genome hasn't been annotated to other like LSW or LW certain genes, so we don't know whether we can further diversification of the photoreceptors, but the information I get from Tom actually is other lamprey species that has more photoreceptor type. Like they even can have multiple different cone type with different
In terms of
opsin
opsins.
genes
Mm-hmm.
expressions. This could suggest that like this horizontal cell could form a different circuit, depends on what kind of the photoreceptor they want to connect.
Right.
But it's pure my speculation.
One question that I have in terms of bipolar cells this time. So, if I'm not mistaken, a couple of years back, there was a paper from Gordon and Sam finding in lampreys a rod bipolar cell that was not on, it was off.
Yes.
And I was wondering whether, when you look at the molecular signatures of the off
Yeah.
bipolar cells, if you can like find this type and how it compares with on bipolar cells in other species.
Yeah, so actually I didn't mention because of the time restriction. We did find one could potentially be this kind of so-called off-rod bipolar cell, is they do share some molecular markers with rod bipolar cells, which we speculate is they could potentially have the off bipolar cell already determined by kind of some shared molecular features with some classical rod bipolar cell. So we think this is very possible.
Right. Thank you very much. And one last question from my side before we move things offline with anyone that might be interested. So I was wondering, because at the last part of your talk, you also talked about development in primates. And again, if I'm not mistaken, because I don't know much about lampreys, there is a stepwise development in their retina. So the larval phase of the lamprey, I think, has only the vertical excitatory pathway, like photoreceptors, bipolar cells, and ganglions. And during metamorphosis, it develops amacrine cells and horizontals. And I was wondering whether you're considering doing some transcriptomics at the larval stage to see how the profiles would be there.
Yeah, so I think some labs kind of trying to take this lead. I think Joe Colby lab from the WashU, they may really kind of starting to really looking at what's the neuron genesis from the lamprey during the metamorphosis. So I think one thing is, I think maybe it's not very accurate. I think the larva stage is largely is the progenitor cell. They have certain like rods, photoreceptor, bipolar cell RGC in the most center part of retina, but the largely is the neural progenitor cells in the larva stage. So actually, most of the cell type is generated through the metamorphosis. So this is kind of recapturate the thing of what we see in the other jeweled species, but they kind of retard or delayed in certain stage, because their environment or their habit don't need a larva stage. So that's
I see.
also make people think the lamprey is kind of primitive vertebrate because they have this kind of weird larva stage. But it's really controversial because some recent evolutionary discovery is they find like in ancient lamprey does not have the larva stage. It's called make it the evolution more kind of controversial, or what do we think about the lamprey? Could we study the modern lamprey, or what really make this cell type diversification? So still, it is all the evolution study. It's based on what the evidence we have, but likely will change if we have new evidence.
Right. And I understand that we don't have time and money to study every retina out there, but I think Lamprey is a good candidate. Okay, so at this point, I would like to thank you once again for this lovely talk, Yirong. And given that there are already people gathering in the Zoom room, I think we should take this offline. So thank you again, and thanks to the audience as well.
Thank you.